So the second speaker in the session today is Kevin JAHNKE, and he is a PhD student in the Göpri group and at the Max Planck Institute for Medical Research in Heidelberg. So Kevin, welcome.
Thank you very much. Just let me share my screen. Okay, this should work now. Yeah, so hello everybody. My name is Kevin JAHNKE. As Darko said, I'm a PhD student at the Max Planck Institute for Medical Research in the group of Kerstin Göpri. And today I actually want to talk to you or tell you how we can actually merge the capacities of top-down and bottom-up synthetic biology to engineer a rather complex signaling pathway in which proton gradients from light harvesting E. coli control DNA assemblies for synthetic cells. And in general, in bottom-up synthetic biology, we try to build a minimal cell module to study or even re-engineer basic biology. And the idea is then to little by little increase the complexity by combining many different of these individual modules to then in the end up with a synthetic cell. And towards this aim, we actually also developed already quite a few dynamic systems in which we can, for example, induce the targeted ectomyosis and contraction inside cell-size compartments using DNA and upon light stimulus, which you can see here in this video. But we can also, for example, engineer the motility of actin containing cell-size droplets, which you can see in this video here. In this case, it's a rotational motility, but we can also change the setup to induce a translational motility of these cell-size droplets. And finally, we can, for example, also look at the division of giant uniramar vesicles upon a light stimulus. One example I show you here, where we actually have a DNA-containing giant uniramar vesicle, which is dividing upon light illumination. However, as you might already see from all of these systems, there are still a few drawbacks. So on the one hand, we still rely on natural proteins that make up the cytoskeleton. In this case, it's actually actin. And secondly, we don't have an energy regenerating system. So once the energy is released, it's actually gone. And therefore, we actually pose the question if we can, for example, build a DNA-based cytoskeleton, which also, and a system which also includes an energy regenerating system. And the problem is, if we stay in the bottom-up approach, this is actually rather difficult because we still rely on the purification of proteins. Many buffers are incompatible with each other. And there's lots of crosstalk. And additionally, the efficiency of reconstituted proteins is often much lower than they are in the natural state. And therefore, there's a different approach to synthetic biology, which is the top-down one. And here we actually start with a natural cell, and we start by reprogramming it with different functions. And the idea is then to take apart the unessential components little by little to then end up with a minimal synthetic cell module. However, also here we have a problem because we don't really know all the essential components that are necessary to build actually one of these minimal synthetic cells. So taking them apart is very hard if you don't know what you're looking for. And therefore, we actually tried to merge the advantages and capacities of these two approaches to, in the end, engineer a more complex synthetic cell pathway than one of these two approaches could do on its own. Okay, so in order to get an energy regenerating system, we actually started off by top-down engineering genetically modified E. coli, which you can see here in this sketch. And these E. coli are special in a way that they actually overexpress a proton pump, which is called xenorhodopsin. And the cool thing about xenorhodopsin is actually that upon a light stimulus, so you just illuminate the E. coli solution with light, it induces a proton gradient across the membrane and thereby acidifies the E. coli interior and basifies the outer aqueous solution here. And this is something we can also verify with pH electrode measurements. So this is just about measurement of the outer aqueous solution when we put an epi of these E. coli into a pH meter. And what you see here is actually that only upon a light stimulus, the pH is actually increasing by about 0.8 from 6.2 to 7.0. And once we turn off the light, it's actually dropping back due to dissipation of protons, and then we can start the whole thing again. So this way, we actually have a rather nice energy regenerating system, which we can trigger by light illumination. As a next step, we actually went on and encapsulated these genetically modified E. coli into surfactant-stabilized water and oil droplets using droplet-based microfluidics. One exemplary image you can see here on the bottom left. And then we actually try to reconstitute the same light-responsive behavior inside cell-size compartments. In this case, we use the pH-sensitive dye pyranine, so a fluorescence-based readout, since we cannot go with a pH electrode into these water and oil droplets here. And what you can see in this plot is actually that we can reconstitute the same light-responsive pH behavior. So only if we illuminate our droplets, the pH of the solution is actually increasing, and then it's dropping down over time again. So what you've seen here is actually that the E. coli act as light-activated synthetic organelles that change the pH inside our inside compartments. And now the question arises how to actually make use of these proton gradients. And since we're a group working on DNA nanotechnology, we actually use the rather common triplex motif, which is just a pH-sensitive motif, which upon basification of the solution is opening up. So these Hochstein base pairs here are actually destabilized, and thereby we have the triplex in this open conformation here, in which it can actually undergo a strand displacement reaction with a partially complementary DNA, and thereby form this duplex here. And now you might wonder why the single-stranded DNA here actually contains a cholesterol modification. And the reason for this is actually that we found that cholesterol-tagged single-stranded DNA molecules are actually inserting at the droplet periphery of our surfactant-stabilized droplets, and thereby act as a recruitable handle there. So in this way, by using the system, we can actually induce the localization of the triplex molecules to the periphery by changing the pH. So at low pH values, the triplex will just stay in the aqueous lumen, whereas at higher pH values, it will open up and then bind to the droplet periphery in this way. And this is what you can also see from these confocal images here. So at low pH values, the triplex is just homogeneously distributed in the droplet lumen. And the higher we go, the more it's actually binding to the droplet periphery, having a turning point here at around pH 6. And now we made an interesting observation, because if we put fluorophore modification on the single-stranded DNA here, just for visualization purposes and verification, we actually found that this equilibrium state here is shifted towards higher pH values. So what has been pH 6 before, now if we have a Cy3 modification on the single-stranded DNA, is actually now at pH 6.5. So what we see here is actually that the fluorophore makes a difference on the conformation of our DNA nanostructures, which was really striking. And we tried to pursue this further by looking at MD simulations of single-stranded DNA oligos. And I want to show you two here. One is an MD simulation of unlabeled single-stranded DNA, and one is the simulation of the same DNA containing a Cy3 modification. And what you see in these videos is actually that the one with the Cy3 modification is much more likely to be found in this rather compact conformation that you can see here, in which it is less accessible for duplex formation. And this, together with dynamic detachment experiments, actually led us to the conclusion that, on the one hand, the unbound single-stranded DNA state is actually stabilized by the presence of a dye, but also once the complex is formed of single-stranded DNA and triplex, this is also stabilized by the presence of a dye, leading to this free energy sketch that you can see here. And we even went one step further by testing 12 different combinations of fluorophores, either on the single-stranded DNA or on the triplex DNA. And just the take-home message from this year is that the fluorophore modification actually has a dramatic impact on the binding of our triplex molecules to the droplet periphery. So there are some combinations in which the binding is not inhibited at all, and then there are others in which the binding is completely inhibited. Okay, But now coming back to our E. coli-based system, the obvious idea was now to combine the genetically modified E. coli and our DNA-based system, in which actually the pH makes difference on the conformation of our DNA by recruiting it to the droplet periphery. And here in this confocal time series, you can actually see water in all droplets containing genetically modified E. coli and the triplex DNA and the control-tag DNA. And what you actually see is that over time, the DNA, the triplex, is actually opening up and binding towards the droplet periphery. And the nice thing is that this only happens upon light illumination. So we actually have proof that this is due to the basification of the outer aqueous solution due to the proton gradients from the E. coli. And now the interesting thing here is actually that once the DNA, the triplex, is localized at the droplet periphery, it doesn't go back. So it just stays there. Whereas we know that the proton gradients from the E. coli actually are reversible. And this was pretty interesting. And what we found here is actually that the small gradients from the E. coli system, which are about 0.8 in delta pH, are not sufficiently high enough to lead to the detachment of the DNA from the droplet periphery, whereas higher pH gradients actually manage to detach the triplex also from the periphery. So what I showed you here is actually that the E. coli mediate the DNA attachment to the compartment periphery. However, that this is only reversible for high pH gradients and therefore not with our E. coli-based system. And now once we can actually attach the triplex DNA via our E. coli, the question arises how to achieve a more meaningful function. And in this regard, we now actually designed a much more rigid DNA-based structure, namely DNA origami, which we actually modified at 4 positions with the triplex molecule, thereby making it its attachment to compartments pH sensitive. And the DNA origami is about like 50 times 50 nanometers, which you can see here. It's a 2-layer sheet. And additionally, we designed the DNA origami in a way that it's actually able to undergo blunt end stacking interactions on all of its 4 sides. And for this, we just need to add the edge staples at the scaffold seam here, which then induce the blunt end stacking. And how this looks like, you can see here from these AFM images. So if we omit the stacking interactions, you will actually see the DNA origami monomers loosely distributed on the mica surface here. Whereas if we induce the stacking by adding the staple strands, we see these huge micron-sized clusters of DNA origami. And what happens actually if we attach these clusters now to giant human vesicles, so by moving also to a physiologically more relevant system, is actually that you see these dramatic deformations and morphology changes of our initially spherical giant human vesicles. So what you see here is the lipid channel at elevated pH, and here you see the DNA origami. And what is striking are these really long, straight kinks and boundaries, which are presumably these huge micron-sized clusters, which just form these flat sheets on the membrane here. And the nice thing about our DNA origami-based system is actually that it's pH sensitive. So what we can do is we can just lower the pH by adding an acid. And what we see is that the DNA origami will detach from the GOV periphery, and our GOVs will actually go back into their spherical shape. And a different phenotype, I would say, compared to this deformation, which we actually observed, is the suppression of membrane fluctuations. So what you see in these videos here is once just a deflated plain GOV, not containing any DNA origami, which is wobbling around. And then on the right, you see a DNA origami covered GOV, which is very, very static, and you don't see any membrane fluctuation, nearly no membrane fluctuations at all. So we also see that we actually change the compartment mechanics via our DNA origami. Okay, so lastly, the aim was to now combine our DNA origami-based systems together with our genetically modified E. coli. And we managed this in a 2-step process. And the first process was actually that we immersed our GOVs in an E. coli containing solution, which also contained the DNA origami monomers. And then by illuminating our solution here, we actually induce the DNA origami attachment to the GOVs. And this is what you see here in this video, which is also verified here in this plot. So what you see here is actually that only upon light illumination, the DNA origami signal at the GOV periphery is actually increasing. And the nice thing, since this is light mediated, we can even do this sequentially. So after waiting some time, we can just illuminate our sample again and then induce more DNA origami to attach to the GOV until a certain threshold is actually reached here. Okay, so now we have the DNA origami monomers on the GOV surface. And then in the second step, we actually need to induce the DNA origami polymerization on our membrane. And this is done by just adding the staple strands. And here you just see a GOV containing or surrounded by genetically modified E. coli. And now the solution contains the staple strands. And what you see is actually here again the formation of these large kinks, which correspond to the DNA origami polymerization on the GOV membrane. So what you can see here is actually that proton gradients from light harvesting E. coli mediate a DNA origami-induced morphology change of our giant unilunar vesicles. And therefore, to summarize, what I've just showed you is that, or how we can actually merge the capacities and advantages of bottom-up and top-down synthetic biology by engineering a complex signaling pathway in which light illumination of our sample or of the genetically modified E. coli actually leads to a pH increase of the outer solution. This in turn causes the triplex to open up and making the DNA origami monomers attached to the GOVs. And then by adding the staple strands, we actually induce the DNA origami polymerization on the GOV membrane, which in turn changes the compartment mechanics and morphology. And with this, I'm at the end of my talk, and I would like to acknowledge the many people that actually helped during this project here. And I thank you for your for listening to me, and I'm very happy to answer any questions you might have. Thank you.
Thank you very much, Kevin, for presenting this impressive work. I'll start with a question. It's as follows. Several diagnostic methods use strand displacement reactions and computation with fluorophores and quenchers for visualization. Have you tested if the most used fluorophores, Tamara, Texas, etc., have a strong effect in single-stranded DNA strand displacement reactions?
So the fluorophores we use, we did not use Texas Red in this case, but we were mainly based on Cy3 and Cy5, so cyanin dyes, and additionally we were testing a few Atto and Alexa compounds, because I mean, if you look at the structures and the charges, they are very different. And for us, it was just important to get a little bit of everything. So some that are positively charged, some that are negatively charged, and some bigger ones, some smaller ones. So we did not study, for example, FITS or Texas Red. But I would assume that they have an impact on the conformation.
Thank you. Have you probed the integrity of the phospholipid membrane after the DNA origami have attached to it? Do the membranes become porous?
Yes, this was also a question we had actually, and we addressed this via FRAP. So we did FRAP of the lipids and the DNA origami, and you see that's the same diffusion coefficient before and after, and even of polymerized DNA origami and just loosely distributed DNA origami monomers. So the lipids and the membrane is actually intact in this case.
Thank you. Have you been able to characterize the sizes of the patches of DNA origami? That is, how many origami combined together?
So I assume the question, so on the GOV membrane, this is hardly feasible because I mean, we don't see the individual origami monomers. And in AFM studies, we're not looking at the discrete size of the patches I just showed you, but we were more looking at the correlation, for example, of different DNA origami monomers and their orientation. So this we definitely looked at. But for example, we did not classify the different cluster sizes because we also assume that they are slightly different on the GOV membrane rather than on the mica surface in the AFM.
Right. Thank you very much.
Sure.
So that we've come to the end of the questions. So thank you very much, Kevin.
Thank you.
And the slide currently shown is the invitation for next week, which is the final session of the speaker series of the SynCell conference. Once again, please register for the main conference event, which will be May the 18th through the 20th. Thank you.